For those who don't know me, my name is Marta Kohmann. I am postdoctoral associate here at the Range Cattle Research and Education Center, working on the long-term agroecosystem research project. Just so everybody who is watching online know, you can type questions throughout the presentation, and I'll start answering them at the very end. So the title of my presentation today is Assessing the Effect of Prescribed Fire Frequency and Mechanical Treatment on the Sustainability of Southern Florida Rangelands. So this is an outline of my presentation. I'm going to start talking a little bit about what it means being a part of the long-term agroecosystem research team, which is, so LTAR, you're going to see me refer to this project as LTAR project throughout this presentation, and that stands for Long-Term Agroecosystem Research. And I'm going to talk a little bit, give an overview about this network. I'll give some details about what it means to be a joint LTAR location with our collaborators at Archbold Biological Station. And then I'm going to talk a little bit more about the native flatwood rangeland, why it is so important, what is a part of our project, and why we decided to use fire and mechanical control. I'm going to present the treatments that we're imposing and talk a little bit about some of the measurements that we're doing. So I'm going to talk about the temperature that we measure during fire. I'm going to talk about, in terms of ashes, how much ashes are deposited during fire events. Talk about vegetation collection and about changes in soil, carbon, nitrogen, phosphorus, and potassium. And then I'm going to finalize this presentation going over some of the other ongoing efforts related to this project. So who is a part of the RCREC LTAR team? This is a very large project. So other than me, all the faculty members here at the station are a part of this project. So Dr. Maria Silveira, who is the guide of the project, my direct supervisor, Dr. Raul Botten, Brent Sellers, Joe Vandermeer, and Felipe Moriel. We also have students. We recently welcomed Shanna Stingo, who is a master's degree student. She just started her program here at ONA. We also have a PhD student who is on campus, Deep High. And we have Vinicius Gomez, who is a PhD student in Brazil, who is a visiting scholar who has collaborated with us in terms of sample collection and sample processing. Some of the other research assistants who you might remember who have been a part of this team, Carolina Brandani, Britt Smith, Casey Okama, Lucas Zanini, and Igor Machado. So all of these people have contributed into the planning and execution of the experiment that I'm going to be talking about today. So what is the USDA Long-Term Agroecosystem Research, or LTAR? So this is a network that was established in 2011 and has been dedicated to develop long-term transdisciplinary research that focuses on increasing the understanding and also forecasting the capacity of the United States to provide agricultural commodities and ecosystem goods and services under changing environment and resource use conditions. So as a network, this is aiming at increasing basically sustainability in agricultural production. And you can see here in this map all the different 18 locations. And they're classified by what is their main focus in terms of work. So in green, you can see the areas that are dedicated mostly to cropland. In that tan brownish color, that's grazing land. And there are some that are integrated systems. Those are in blue. So in the Archbold Range Cattle Research and Education Center is a joint location. And we entered this network in 2014 through a competitive process. And we are one of only 2 of these units that are not USDA. This network is separated in working groups. So what are these working groups? So across this large network, the scientists that are a part of it are grouped within common interests. So for example, we have a soil health working group, a grazing lands working group, and so on and so forth. There are more than 20 different groups. And the objective is to kind of pair up different scientists who are across these locations who have common interests. And what that also means is that the research that we develop here in Florida has more impact because it's shared and compared to all these other different locations. So there are different protocols that have been established that are repeated across this entire network, all these different locations. So talking a little bit more about our joint location, which is the Archibald Biological Station and Range Cattle Research and Education Center together. So this map here is showing the cattle inventory by county in Florida, and these two circles are showing the locations of our two experimental sites. The first thing that I would like to emphasize is that we are located where the beef industry or the cattle industry is really important in terms of economics, right? And some of the characteristics for those who are not familiar with the beef industry, more particular here, is that a lot of the operations are cow-calf operations, and they mostly utilize forage as a source of feed for the animals. That varies between cultivated pastures and also native rangelands in more extensive production systems. And in this presentation, I'm gonna talk mostly about the native rangelands. Okay, so why are the native rangelands important? This picture here is showing a typical vegetation formation. So what we have is an overstory of some oak trees, some pine trees that are kind of scattered throughout the landscape. We have a lot of saw palmetto plants, and then on the understory, we have mostly C4 grasses. In terms of this ecosystem, why it is so important, in terms of biodiversity, this ecosystem is very rich. There are more than 300 plant species in this environment, although around 15 of them, so 15 of them make the majority of the biomass. So one of the most important that I think everyone who is from South Florida is familiar with is saw palmetto. So looking just at saw palmetto, we found in the literature that more than 300 pollinator species visit the flowers to collect pollen. And we know that saw palmetto, both the fruit, the fronds, and the heart of the palmetto are part of the diet of many different animal species. So from black bear to raccoons and white deer. And it has also been documented that the Florida panther makes their dens on palmetto plants or close to palmetto plants. However, an important aspect of this ecosystem is that it's one of the most threatened. And it's estimated that around 64% of the area is already lost, and this is mostly due to urban and suburban areas that grow into these areas. Still looking at these native rangelands in terms of the importance for the beef industry, a lot of producers will have both cultivated pastures and native rangelands kind of intertwined in their areas, in their production areas. They're mostly used for dry pregnant beef cows during the winter. And some of the characteristics of this environment is that it produces a lot of biomass, and because of the high incidence of lightning in Florida, typically fire occurs every 3 to 4 years, and this is a natural occurrence, okay? So with that in mind, the land managers use prescribed burning typically as every 4 years, so at the close to what typically occurs in nature, in order to, one, try to reduce a little bit the presence of palmetto plants and increase forage production on the understory, and two, also trying to reduce the danger of occurrence of uncontrolled fires in their areas. And then some producers will also associate chopping either before or after fire to really try to hurt those saw palmetto plants and really reduce their biomass in their areas. So knowing all of this, we delineated an experiment with 4 treatments. The first treatment is the fire every 4 years. So across the LTAR network, typically each one of the locations has a business as usual treatment. So this encompasses the typical management practices of whatever it is that they work with, whether it is soybean, corn, whatever it is. So for us who are working with native rangeland production, our business as usual, our typical is the use of fire every 4 years. The second treatment, which we call aspirational, so it's the one that we want to see if they improve the different measurements that we're taking, is the fire every 2 years. So we're increasing fire frequency. We also have a fire every 4 years with mechanical control, which is done with the broiler chopper. And we have an unburned treatment. This unburned treatment is a control, so we can compare also all the other treatments that have fire mechanical control to this one. And we just started imposing these treatments in 2019. And you can see here in this image that we have some animals here, right? So our aim is to put animals to graze, or this is what we're going to do. We're going to put animals to graze between September and February of every year. We're going to use dry cows that have a lesser nutritional need. And we're going to weigh the animals and give a body score to the animals before and prior they leave the experiment. And this is going to be the same grazing management across all the different treatments. So let's take a look at our experimental area. So this is a map of the RCREC. To knowledge, Bethany actually constructed these maps for us. So what we can see here is that we have 4 different blocks, and each one of these 4 blocks has 4 plots because we have 4 treatments. So we have a total of 16 plots, and each one of them has between 40 and 60 acres. So they're very large. This means that almost half of the area of the research station is dedicated for this experiment. And to our knowledge, this is the first time that an experiment this big in terms of area and also aiming at looking at long-term effect of a treatment has been done in this kind of environment. So this is really important research that we're developing. I also want to point out that in each one of these plots, you can see these little yellow lines. Let me point them out right here. So each one of these 16 plots has 5 delineated transects. These are vegetation transects, and they are around 150 feet long. So the purpose of having these transects in these plots is that we go back always to the area between these transects to do our sampling, because our experimental area is very large, so there is a lot of spatial variability. So we go back to the same transects to do the measurements to try and reduce the variability we see a little bit. So just so you have an idea of how this vegetation looks like, this is an unburned area. Just so you have a reference, this is me on the picture in the orange shirt, and I am 5'3". And this is an image of the burned area. This picture was taken immediately after fire, so you can see that you can still see some of the structure of the palmetto plants, but the biomass is pretty much gone, right, in terms of the green part. And then this is the area that was chopped after it was burned. And you can see here that there is some green in the area. This is because between burning and chopping, there was around a couple of months. So the vegetation had a little bit of time to bounce back before it was chopped. So this is why you see this green here. All right, so looking at our treatments, I'm gonna go over some of the measurements that we took this year. So as I mentioned, we introduced fire and chopping this year, and the fire occurred between late February, mid-May of 2019, and then the areas that were gonna be chopped were chopped between May and June. Okay, so let's look at temperature during fire. We measure temperature during fire using thermocouples, which is this that you can see against my glove here. This part of the wire that is exposed is the only part that stays on the soil surface that actually measures the temperature during fire. This wire is connected to this data logger that is collecting data every 10 seconds. So this part of the wire and the data logger are actually buried between 6 and 8 inches below ground to protect the device against the temperature. So the reason why we wanted to measure temperature is because it can be an important determinant of how much carbon and how much nutrients we're losing during fire. That can also have some impacts on the soil microorganisms. So when we are looking at the average temperature during the peak, so during the peak temperature, we had an average temperature of 500 Fahrenheit, but the range of peak temperature was really, really large. So the minimum temperature that we recorded was 120 degrees Fahrenheit, and the maximum was over 1,000 degrees Fahrenheit. And this graph here is showing the peak temperature on the y-axis and the temperature average at each one of the blocks. And you can see that it's pretty variable, right? And this variability was probably due to some weather conditions and vegetation. So perhaps this year was not the absolutely ideal year to start imposing fire because we had an unusually wet winter and wet spring, and that caused a lot of spatial variability in terms of humidity in the soil, and that affected the temperature, the peak temperature of the fire quite a bit. So how variable is this? Let's take a look at this unit here. So this is Woods 2, Unit 2. If you see here in this picture, we have the 5 different transects in this unit. So this is transect A, this is transect C. And this graph here is showing the temperature in Fahrenheit in the y-axis and the time of the day in the x-axis. So this is, we installed the thermocouples in 3 transects per experimental unit, either the day before or right before the fire was imposed. So this is throughout the day that we imposed fire, how the temperature was varying on soil surface. So you can see here, this is when the fire went by, that the peak was around 300 degrees Fahrenheit, right? And this is in transect C. So when we look at transect A, the peak was 900 degrees Fahrenheit, and these 2 transects are around 150 feet away, so they're not that far apart from each other. And one of the also, sorry, before I move on, I also want to emphasize how short these peaks are. So within 2-3 minutes, the temperature will go to 100 to the peak and go back down. So these peaks are really intense, but also very short-lived. So this picture he's showing the vegetation. This was actually afterburn of the transect A and the transect C. We can see here there are a lot more palmetto plants on transect A relative to transect C. And the palmetto has some oils in the plant that really help with the burning. So what we think is happening is that the variation in vegetation is really affecting how the fire behaves, how the temperature behaves during fire. Moving on to the ash collection. So just for the purpose of this presentation, when I talk about ashes, I'm talking about whatever biomass burn that was deposited or existing on the ground. So after a fire went by, what was still there in terms of burn material. And the reason why we're interested in measuring ashes is because it can be a source of carbon and also a source of nutrients to the ecosystem. And we did measure ashes in 2 different ways. The first one was what we call wet collection. So basically, when we're going outside to the field to install our thermocouples to measure temperature, we also installed these aluminum trays on the soil surface. So we dig the soil a little bit to make sure that the top of the tray was right at the surface of the soil, and we fill that out with water and put 2 of them per transect. And the idea is that during the fire event, as the ashes, as the biomass is burning, some of the ashes are being deposited on soil surface are going to be caught on these water traps. The second measurement that we did was after fire. So we're calling that a dry collection because after the fire ended, we went out there and used this circle and collected a couple of them per transect. And this is just so you can see the ashes here on the soil surface. This is where we had just collected material. So it's the material remaining after the fire. In terms of how much was deposited, From the wet collection, we found around 400 pounds of ashes deposited, and from the dry collection, almost 1,700 pounds per acre. In terms of nutrients, so we analyzed this for carbon, nitrogen, phosphorus, and potassium. In terms of nutrients, this represented more than 500 pounds of carbon per acre, 13 pounds of nitrogen, and around 3 pounds for phosphorus and potassium. So quite a bit. Moving on to our vegetation collection. So again, this is a picture of the vegetation before burning. And this picture was taken around 4 months later in an area that was burned and chopped. And if you remember from the beginning of my presentation, we know that land managers use prescribed burning fire typically every 4 years, and some of them associate that with chopping. And most of the objectives is to try and reduce the presence of palmetto plants and also increase the presence of some of the more desirable plant species. So what we wanted to know was, how will these treatments affect vegetation, biomass and vegetation composition? So we went out to sample these areas in September. We sampled around 25 square feet per transect. And there are 80 transects in this experiment. So in between 3 and 4 people, it took us 1 month to finalize the sampling in this entire area. And we wanted to quantify total biomass, and we also wanted to see how this biomass was partitioned between different functional groups. So we're interested in looking at saw palmetto plants, desirable and undesirable grasses, forbs, and shrubs. So for those who are not familiar, this is a palmetto plant. We collected that the whole front, so all this green part here, and that was collected. The other vegetation was collected to ground level. So in terms of desirable grasses, these are some of the plant species that we consider desirable. Some of you may be familiar with these plant species being classified as decreasers because they typically decrease in presence in unfavorable management conditions. And they're classified like that in terms of persistence, but also in terms of animal preference. So these plant species that are listed here are preferred by animals, and also if you mismanage your rangeland, they're going to reduce in presence. In terms of undesirable grasses, these are some of the ones that we found. So wiregrass, Indian cup scale, carpet grass. We found a lot of different forest species, including legume plants like the wild sensitive plant and sedge. In terms of shrubs, we found blueberry, blackberry, and gallberries quite a bit. So let's take a look at biomass production. So this graph here is showing unburned, burn treatment. So we grouped the 4 and the 2 year burning together here, and the burn and chopped. And this is showing biomass in pounds per acre. So what we found here is that the total biomass was similar between unburned and burn treatments. You may be wondering, how is this similar in any way, right? These pictures that I showed you early in this presentation. So one of the things that happened is that because we imposed the fire between late February and May, and we only sample vegetation in September, this environment that has evolved with fire, the vegetation just really bounced back. So this is why you see a similar biomass production between the two treatments. When we look at the biomass in the areas that were burned and chopped, we see that there was a reduction in around 45% of the biomass. But we also wanted to look at biomass distribution. So how about the distribution among the different functional groups? So these graphs here are going to show the percentage biomass of each one of the categories that's going to be on the x-axis. And the blue is showing unburned, orange is showing burned, and the gray is there that was burned and chopped. So when we look at the palmetto proportion, we see that there is a greater proportion of palmetto plants in the unburned treatment. So burning and chopping or just burning reduces the presence of palmetto plants. In terms of desirable grasses, the burn treatments have the greater proportion of desirable grasses that was lower in the unburned treatment. In terms of forbs, we can see that the areas that were chopped had a greater proportion of forbs. Shrubs were very variable. There was no difference in the just numerical difference, no statistical differences among the different treatments. There's a little bit greater amount of undesirable grasses also in the chopped area. So from this data, what we see is that the unburned areas have more saw palmetto and less desirable grasses. And I brought these pictures here. These are 4 different transects across different units that were not burned that we collected. And you can really see mostly palmetto plants in these pictures. When we look at the burn treatments, there's less saw palmetto and more desirable grasses. And again, I brought some pictures here of the burn treatment. And we can see relative to the unburned, there's a lot less palmetto plants. And we can see some of the desirable grasses here, like chalky blue stem, just this one here. And finally, when we're looking at the areas that were both burned and chopped, there's less saw palmetto, but also there's quite an increase in the presence of forbs. So when we're looking at the pictures here, especially this one, you can see there's less saw palmetto, but you can see a lot of sedge, a lot of dogfin, a lot of other forbs in this treatment. Moving on to some of the soil chemical characteristics. So I'm going to be showing some data of pre-fire and post-fire in terms of 2 depths, so 0 to 4 inches and 4 to 8 inches. So these pre-fire samples were taken around 2 months before, 2-3 months before in post-fire. And the post-fire samples that I'm presenting here were taken immediately after fire, so the day off or the day after fire. So here, these lowercase letters are showing the difference between the same time of collection, so pre-fire or post-fire, at the 2 different depths. So there is more carbon in the upper 4 inches of the soil in the pre-fire, and this is also true for the samples taken post-fire. Now, let's compare pre and post-fire. So this is the uppercase letters. So we didn't see any effect of fire in carbon, total soil carbon concentration before or after fire. Looking at nitrogen, again, the lowercase letters is comparing between the 2 depths at the same time of collection. So the 2 blue columns, so the pre-fire, again, there's more nitrogen concentration in the top 4 inches of the soil and less in the 4 to 8 inches. And although there is not an effect of fire here on the first 4 inches of the soil, we see that after fire, there was an increase of nitrogen concentration in 4 to 8 inches. Looking at extractable phosphorus, we see that after fire, so the uppercase letters, there was a reduction of extractable phosphorus in the soil from pre to post fire. And in terms of potassium at the 4 to 8 inches, there was an increase of extractable potassium in the soil. So other researchers have found in different environments that there are some short-term increases, especially in soil extractable nitrogen. And this is probably due to inputs from ashes. So if you remember from the slides that I showed about ashes deposition, we do have some nutrient inputs from the ashes after a fire event. And when we look at the phosphorus here, what we think might be happening is that this is not total soil phosphorus, it's extractable soil phosphorus. So what could be happening is that the availability of that phosphorus decreases after the fire. So we are still, this is a hypothesis that we are still investigating. Okay, so to finalize this presentation, one thing that I really want to emphasize is that this is just a glimpse over what we're doing in terms of this experiment. So I only presented a short amount of the data that we are collecting. We have a lot of ongoing studies. So we have some studies on nutrient cycling where we're investigating above ground litter decomposition, comparing, looking at how burning affects the composition in terms of botanical composition, interacting with nitrogen application. We're also looking at a lot of soil responses in terms of soil carbon stocks and stability of that carbon after fire. we're looking at natural abundance of carbon-13 in an effort to allocate or to determine what is the source of this carbon being incorporated in the system. Our master's degree student, Shona Stingo, is looking at impacts of pyrogenic carbon on soil carbon stocks and long-term stability. As I mentioned, there is a PhD student who is housed on campus, Dipti Hai, who is looking at soil microbial community responses to fire, looking at both diversity and activity and soil enzymes and RNA. We're also doing greenhouse gas evaluation of burn and burn and chop treatments. And we have some studies that are comparing different response variables in terms of greenhouse gas emissions and nutrient cycling, comparing cultivated pasture to native rangeland. One of the other measurements, some of the other measurements that we're doing, one important aspect of this network is that, as I mentioned, there are some common protocols that are installed across the network. So one of the things that we are measuring that are being measured in other research stations is carbon fluxes, CO2 fluxes through an eddy covariance tower, which is pictured here. And just to mention some of the measurements that this eddy covariance tower takes, other than CO2 fluxes, measuring temperature, wind speed, wind direction, precipitation, and others. We also have a phenocam here, which is basically set up to measure how green a canopy is, and it does that every 30 minutes. So this graph here is showing like a measurement of greenness with time. So you can see that there is some variation in greenness with the seasonality, and then there is a reduction here, right? So what happened here in 2009 to reduce the greenness of the canopy? Fire. This is picking up very nicely, and then there is a growth of vegetation afterwards. Before I finalize this presentation, just want to make sure that I voice our appreciation to all the help that we've got from the forestry department and all the help from the farm crew with setting up lanes, fire lanes, and helping us with fire management, and also installing all the water troughs, etc., for the cattle. So yeah, we're very appreciative of all the help that we got. So with that, I'm concluding my presentation, and I'll be happy to take any questions. So I'll start taking some questions from the audience here, and then I'm going to take a look at the questions online. Questions or comments? No, that's a good question. So the question was if there was any concern relative to displacing wild animals. So we didn't assess really the population, but I'll tell you that like very shortly after the fire, I actually went out there to collect some samples, and we saw, you know, walking among the ashes, some gopher tortoises, and they were just going around their lives. So I am not a wildlife specialist, but I think that at least some of the species they have some mechanisms to cope with the fire. I have a question here online. So Dr. Joe Sanchez is asking if we're evaluating soil pH and if we have any expected responses. So we are measuring soil pH with time. I'm actually not sure if do we have any expectations in terms of changes. I'm looking at Dr. Silveira. Yeah, so Dr. Silveira said that the soil is very acidic. I think the pH range was between 4 and 4.2, 4.5 in these areas. So we're not expecting a lot of change due to fire. Yeah, so the question is the classification of the grasses between desirable and undesirable, if they are based on cattle production or if they take into consideration any wildlife preference. The answer is yes, it's based on cattle preference and cattle management. So yeah, but we do realize we don't want in any way like eliminate plant species from this environment. We want it to be just as rich as possible. It's just trying to skew the production towards something that's more targeted to beef production.